Right. So let us read this question first. Statement 1 is more than one codon can code for a single amino acid. Okay, more than one codon can code for a single amino acid. So you have one amino acid. For one amino acid, you have multiple codons. Right, so this is what is the first statement. The second statement is more than one amino acid can be coded by a single codon. They are saying one codon can code many amino acids, right? So one codon can code many amino acids. This is statement 2. So which statement is true here? Yes, can you tell me which statement is true? Statement 1 or statement 2? Yes, one amino acid has multiple codons. Is this true? Or one codon has many amino acids? Is that true? So statement 1 is going to be true, right? So more than one codon can code for a single amino acid. That is true. But one codon coding for multiple amino acid, it is not true. That's not that. In that case, specificity will be a question mark, right? So that is not true. So which statement is true? Statement 1 is going to be true. Yes, statement 1 is true. Mention the property of genetic code that is applicable to the true statement. So what is the property of genetic code that is applicable here to the statement 1? It is going to be degeneracy. Yes, it is going to be degeneracy. Right. So what is wobble phenomena? Explain with an example. So they've just now in split questions itself, they've given you what is wobble phenomena. In which case, which is supported, which characteristic of genetic code is supported by Wobble's phenomena? It is going to be degeneracy. So what is wobble phenomena? They've asked us to explain with an example, then mention all the properties of genetic code. So this is what is the question. Now let us start from the first basics only, and we will cover what is genetic code. So what is genetic code? Genetic code means itself, it tells you the relationship between the mRNA and the protein sequence, or the DNA, mRNA and the protein sequence. Yes, so from DNA, what you get is the mRNA. And from mRNA, what you get is getting translated to form the protein. So what is the relation? So from the gene is what you get a protein, right? So how do you get this? Yes, and how how this language of mRNA is getting converted to a language of protein. What is the language of protein? It is going to be amino acids. Amino acids which are linked together by polypeptide linkages. That is the language of a protein or a polypeptide. But here in mRNA, it is going to be a sequence of bases, right? It's sequence of nucleotides. So every 3 nucleotide, I will call it as a codon. Okay. So 1, 2, 3, I will call it as codon 1. Then 4, 5, 6, I will call it as codon 2. 7, 8, 9, I will call it as codon 3. Like that, right? So codon 1 will code for one amino acid. Codon 2 will code for one amino acid. Codon 3 will code for one amino acid. Likewise, based on this language of mRNA, codon language, the amino acids are being recruited and amino acids will be linked together to form this polypeptide chain and further the protein. So this is what is the relation from the DNA to mRNA to protein. All right. So that is what is depicted by genetic code. So what is a codon? Codon is a triplet nucleotide sequence that will code for an amino acid. Clear? And the nucleotide sequence of this mRNA molecule will contain a sequence of codons as we saw here. It is going to have a sequence of series of codons, right? So each codon will code for specific amino acids, and that's how many amino acids are getting closer to each other by peptide linkages, and they will form a polypeptide. Now look at this structure. So here is the mRNA, right? Here is the mRNA. mRNA is formed in which direction? From 5' to 3' direction. Yes. So how translation happens with the help of this ribosome in the cytoplasm is where the translation is going to happen. And how is it getting translated? This mRNA is getting translated. So let us see the sequence of this mRNA. It is going to be like this, right? So first 3 nucleotide is going to be AUG. After that, what you have? CUC. After that, GGA. Then UAC. So that's how the codons are read. Okay. So how am I reading? That's how the codons are read. That's why I'm reading like this. Okay, I'm reading it as a sequence of threes, right? So first codon is what is the start codon, and AUG is nothing but the codon that codes for methionine, right? After which you have a sequence of codons. So this CUC will code for one amino acid, then another amino acid, then another amino acid. Likewise, this methionine will bound to the other adjacent amino acids corresponding to the codon, and they will form a peptide bond. So that's what happens in translation. So we will see that in translation. Finally, there is a codon. How will this stop? How will the translation process stop? With the help of a stop codon. Whenever the ribosome encounters a stop codon, the translation will stop. Here the stop codons are UAA, UAG, and UGA. Or if you can, how can you remember this? You are away, you are gone, you go away. Okay, so just remember this, and if it fits this sentence, you can just verify it. Okay, so these three are the stop codons. So how is this codon represented in the mRNA? With the help of 4 bases. So that we know. So you have 4 bases. All right. And how many stop codons are there? 3 stop codons are there. Then 4 bases you have, but the codon is a triplet nucleotide base, triplet nucleotide sequence. So how many different combinations of codon do you have? Yes, how many different combinations can you have? Out of this 4 bases and the codon being read as a triplet sequence, it is 4 to the power 3, which is 64. So I will have 64 codons in me. Out of the 64 codons, 3 codons are stop codons. So you will minus it. So you will have 61 codons that code for the 20 amino acids that you have. Right? That will, 20, that will code for 20 amino acids that you have. So this is what is the basics of your genetic code. So 61 codons are there for coding 20 amino acids. So let us just see this, just for your reference. You can't reproduce this in your exams, but just for your reference, you see what is this, how is this table form, genetic code table form. So here along the x-axis, you will have U, C, A, and G. Then along this axis, you have U, C, A, and G. Then in each table, you write U, C, A, and G. So this much combinations of codons you will get. So U is what? Phenylalanine. So for phenylalanine, how many codons do you have? I have 2 codons for phenylalanine, right? Then for leucine, how many codons do you have? You have 6 codons for leucine. Serine, how many codons do you have? 4 codons for serine. Yes, so look at this. So this is the way you will learn it. So how many isoleucine codons do you have? 3. Then how many methionine codons you have? You have only 1 methionine codon. So you have only 1 codon for methionine. Then how many valine you see, and proline, threonine, alanine? Yes, and glutamine, glycine, arginine. Arginine has many. Again, arginine has 6. This leucine also has 6. So this is how you will see this. And look at the stop codons. These 3 are the stop codons. Okay. And tryptophan, how many codons for tryptophan? 1. So methionine and tryptophan are going to have only 1 codon for them. Okay. And many number of codons are there for leucine and arginine. And you have 3 stop codons. Okay. So this is how you will understand this table. All right. So let us move on to the next. So how, with the help of this, what do you understand? We understand that one amino acid can have multiple codons. Is it not? But one codon will code for only one amino acid. As you see, this U is going to code for only phenylalanine. It is not going to code for any other amino acid, right? Similarly, UAU is going to code for only tyrosine and not any other amino acid. But for tyrosine, you see, you have 2 codons, UAU and UAC. Is it not? How is this possible? That also we will see. So, what are the different characteristics of genetic code here? Degeneracy, yes, you have 5 different characteristics of genetic code. Degeneracy, unambiguous, non-overlapping, non-punctuated, and universal. These are the 5 important characteristics that you need to know. What is degeneracy? As we are seeing in the past few slides, degeneracy means one amino acid can have more than 1 codon. That is what is the degeneracy. So 61 codons will stand for 20 amino acids. So that explains this, right? So, but how is this possible? Yes, how is this possible? You have the mRNA strand here. Yes, so along the mRNA strand, you have the codon. And how is this amino acid getting recruited? So first of all, if I have AUG as my codon, this codon is specific for methionine. So the tRNA that is carrying this methionine amino acid is going to come. But how does it attach to this codon on the mRNA? It will have a anticodon arm. So tRNA has an anticodon arm, right? So that will be complementary to the codon on your mRNA, is it not? So you have an anticodon that is complementary to the codon on the mRNA. So reading this codon on the mRNA, it is going to bring the methionine, and the codon of this, anticodon of this tRNA that's carrying the methionine is going to be complementary to that of the codon on the mRNA. So that's how it is going to get attached, right? So for this, so as you can see, this is the 5' end and this is the 3' end of your mRNA. So you see, this third pair, okay, this first two pairs are very strict. Okay, first two pairs are very strict. The tRNA, you see, this is the 5' and this is the 3'. So the first pair of this, the first bond of this mRNA codon anticodon pairing is going to be very strong. Second also it is going to be strong. But third, when you see, it is going to be weak. Okay, that will be weak. So I can have multiple combinations. It is weak in the sense the Watson and Crick base pairing rule is not being followed that much strictly in the third base pair. Okay, so that is how I can pair with many number of nucleotides. So one nucleotide pairing with another one nucleotide, that is what is very stringent and strict, right? So that rule is being relaxed in the third base pairing rule. That rule is relaxed in the third base pair. So here third base pair alone, it is relaxed. Okay, so that is what is bringing explain the degeneracy, and that relaxation is what is called as the wobbling phenomena. Now let us understand this with the help of an example. Now these are glycine codons. Okay, so here I have an mRNA which is telling which has this GGU, okay, GGC and GGA. So I'm having three codons here on the mRNA. But look at the tRNA that is carrying the glycine. Yes, look at the tRNA that is carrying the glycine. You have CCI. Okay, CCI. Okay, so what does this third pair has? The third one has this, the third pair, this U, C and A are binding with just I. What is this I? I is for inosine. That is what is making this wobble pairing. Okay, so this I can bind with U or C or A. Okay, so just one glycinyl tRNA can bind with three different codons. It is carrying only the glycine. Yes, so for these three codons, I can have glycine only. Understood? So that is how I have one amino acid which can have more than one codon. Okay. Similarly, look at this is one way of wobble pairing. The other way is that is with the help of inosine, you're pairing with multiple nucleotide bases. Similarly, for another way is you see this leucine. Okay, leucine. So CUC, CUU, both of which is coding for leucine. Now, normal pairing means what? The C will bind with G on the tRNA, right? But the U here can bind with G in the tRNA. Yes, so tRNA is nothing but GAG only. But it can bind with both these codons. How is it possible? That's what you should know. Along with the third base pair, there is wobble pairing, which is where the Watson and Crick base pairing rule is not strictly followed. Okay, that is being relaxed there. That is why this can freely bind with other nucleotides even. All right, so this is what is called as the wobbling phenomenon. There's two mechanisms by which wobbling phenomenon can happen. Because of that only explains the degeneracy of the genetic code. Okay, that so because of this, I mean, this is one other reason where reduces the effect of mutations. Okay, so if the amino acid has more than one codon, yes, if the amino acid has more than one codon, the first two bases in the codon will be the same. Only third one is different. This reduces the effect of mutations. Okay, so this is what is about the degeneracy of genetic code, and this explains the wobbling. I mean, this is supported by this wobbling phenomena. So let's just see. This explains the degeneracy of genetic code, decreases the rate of mutation. You have 61 codons as we saw, but how many tRNAs do you have? We have only 31 tRNAs in the cytoplasm. Okay, we have only 31 tRNAs in the cytoplasm. Again, this wobbling phenomenon is explained by this. All right. Then, rule of this base pairing is relaxed at the third position of the codon, so that one base can pair with more than one complementary base in the anticodon. Also, some tRNAs will have inosine. Inosine can pair with U, C, and E. All right. So this is what is about the wobbling phenomena, and with the like glycine and leucine codons, we are able to understand this. Yes. So next we move on to the next characteristic of genetic code. That is specificity. One second, guys. Okay. So next is specificity or unambiguous nature of genetic code. There is no ambiguity in this genetic code. That is, a particular codon will always code for the particular amino acid wherever it is present. So this is what tells us explains the specificity. One codon will code for only one amino acid. One codon will not have multiple amino acids. Okay, so that is what is specificity. Clear. Next is non-overlapping nature. So while reading the codon sequence on the mRNA, while reading the mRNA sequence, how did I read the codons as 1, 2, 3, 4, 5, 6, 7, 8, 9? Is it not? So that's how I read. So that is what is the non-overlapping nature. Okay. So this codon is read from a fixed starting point as a continuous sequence of bases. So as triplet sequence only it is read. So as you look into this picture, ACA is 1. Okay, and TGA, that's how it is read. It is not read like this. Okay, it's not read like this. CAT, then this TGA, then this ATC. It's not going to be read like this. Okay, no overlapping is there. So this is what is about the overlapping nature. Next is non-punctuated. What is non-punctuated? Once the reading is commenced at a specific codon, there is no punctuation. So you don't skip any nucleotide bases in between, right? You don't skip any nucleotide bases in between. So that is what is about your non-punctuated. So again, look at this example, CAT, that's followed by the second codon. If I have any base in between, I will not skip it. Okay, I will not skip it. So 1, 2, 3, 4, 5, 6, 7, 8, 9. That's how it should be read. Right. So next is universal. Universal means what? The codons are the same for the same amino acid in all the species, be it elephant and E. coli, right? So say the codons, for example, AUG is methionine means AUG is methionine everywhere. So that's what they say. But there are exceptions here even. What is the exception in mitochondria? Yes. So let us just see. Spot the difference here. Yes, if you can spot the difference, just leave it in the chat box. So this is the cytoplasm genetic code table. This is the mitochondria genetic code table. So this is how inside the mitochondria also there is the machinery, yes. So that will produce this machinery for protein synthesis. So how this codons are being read. That is what in mitochondria it's a little bit different, and cytoplasm it's what is the universal one. So the exception is found in mitochondria. Can you spot the difference here? Yes, can you spot the difference here? You can just leave in the chat box, right? First is stop codons. In cytoplasm, what is the stop codon that we read? UAA, UAG, and UGA. So there are 3 stop codons. But in mitochondria, 4 stop codons are there. What are they? UAA, UAG, AGA, AGG. These are the 4 stop codons in case of, yes, good, stop codons. So stop codons in, might so keep writing in the chat box. Stop codons 3 in cytoplasm, 4 in mitochondria. All right. Similarly, what is arginine? Where is arginine here? Arginine here is AGA and AGG is arginine in the cytoplasm. But you can see in mitochondria, AGA and AGG is stop codon. Yes, stop codon is what we saw. Next, where is the difference you find? Methionine. Methionine, there is only one codon for methionine in case of cytoplasm, that is AUG. But in mitochondria, you have 2 codons, AUA and AUG in methionine, in mitochondria. Similarly, isoleucine is AUA, but here AUA is methionine in mitochondria. Okay, so that is what these are the differences that you find. So you have 3 stop codons, but this UGA stop codon is what? In mitochondria, UGA is going to be coding for tryptophan. All right, so these are the differences. So what are the differences between cytoplasm and mitochondrial genetic codes? UAA, UAG, UGA is stop codon here, but here UAA, UAG, AGA, AGG is going to be the stop codon in mitochondria. This AGA, AGG is stop codon in mitochondria, but in cytoplasm, it is going to be coding for arginine. UGA is a stop codon in cytoplasm, but is in tryptophan in case of mitochondria. Methionine, you have only one codon in cytoplasm, but you have 2 codons in mitochondria. The other codon that's coding for methionine in mitochondria is going to be coding for isoleucine in cytoplasm. Is it clear? Yes, so these are differences. I guess these are the differences. Yes, any more differences that you find? These are the differences between the cytoplasm and mitochondria. All right. Yes. So one more if you want to know. Tryptophan, only one codon for tryptophan, but you have 2 codons for tryptophan. So in cytoplasm, what did we see? Methionine and tryptophan is going to have only one codon, but here methionine and tryptophan are having 2 codons in mitochondria. Okay. So these are the differences that you can see. Right. Yes, so with this, we are completing genetic code. Right, so hope this is clear. So you have the 5 characteristics. Degeneracy is most often asked because you have wobbling phenomena associated with it, which supports this degeneracy. Also, they may ask you why the mutation, which characteristic of genetic code is protecting you from mutation? That is going to be degeneracy only. Okay, so that is what is about your genetic code. Next, we move on to the next question. So a 55-year-old person was diagnosed with type 2 diabetes. Diabetologist asked him to get HbA1c estimated. His HbA1c was reported with a special mention that there was an abnormal migration of hemoglobin in the electrophoresis. His SpO2 was normal. So what do you think is the answer here? Yes, what do you think is the answer here? Anybody? So first of all, the diabetologist was asking for HbA1c. So usually we do HbA1c in HPLC, right? So HPLC not just gives you HbA1c, but it also gives you hemoglobin variance. But the mode is different. Okay. So the HbA1c was reported and he was doing this hemoglobin electrophoresis also. So hemoglobin electrophoresis, again, you're doing it for abnormal migraine, doing it for finding out the HB variants. Okay, so HB variants only. So what are the different HB variants? You have multiple. So the normal ones are what? HbA is adult hemoglobin, HbF is fetal hemoglobin, then HbA2 fraction you have. Yes. So you also have HbS fraction, HbD, HbE. Okay, so these are abnormal variants of hemoglobin that you have. So they are mentioning that there is abnormal migration of hemoglobin in electrophoresis. So after finding out that HbA1c level he gave HbA1c test only. But what happened? The level of HbA1c would have been more or abnormal. Yes, which did not correlate or something. So that is why he did this HB electrophoresis, where he found the abnormal migration of hemoglobin. So all of you have given good answers. It is a hemoglobinopathy. Okay, it is a hemoglobinopathy only. But what is a hemoglobinopathy? They are mentioning that there is abnormal migration of hemoglobin in electrophoresis. So let us just see this abnormal migration here. Okay, what is this abnormal migration? Normally, this in hemoglobin electrophoresis, if you do on the electrophoretic slide, how are the HB fractions going to move? Okay, normally, how does it move? As you can see, this is the origin of sample application. HbA or the adult hemoglobin should always be more than 80% in normal individual. So you have a broad band here. It is moving to the other end of the electrophoretic slide, right? And you also have HbF fraction that is just before the HbA, and it should be less than 2% normally. And HbA2 fraction you have that should be less than 3.5%. Okay, this is in a normal individual. That's how this is going to be present. When you take beta thalassemia trait or beta thalassemia major, beta thal trait means what? It is comparatively less symptoms are there. Beta thal major, you have severe symptoms where the patient will be more transfusion dependent. Yes. So trait, if you take, the characteristic of beta thal trait that you will find in an electrophoresis is going to be HbA2 fraction, which is more than 3.5. That's what you will find in a beta thal trait in electrophoresis. Beta thal major, if you take, characteristic will be more of fetal hemoglobin. Okay, more of fetal hemoglobin. That is what you will find. And adult hemoglobin is very, very less. That is what you find in beta thalassemia major. Okay. Now look at sickle cell trait and sickle cell anemia. What is sickle cell trait? Sickle cell trait is a heterozygous condition where you have both the abnormal hemoglobin and normal hemoglobin. Normal is HbA, adult hemoglobin. So that is also present. But you have a special fraction which is HbS. Okay, you have a special fraction which is HbS, which is the sickle hemoglobin that will be present. In sickle cell anemia, if you see, there is only HbS. Normal hemoglobin itself is not there. Only abnormal hemoglobin is present. So only HbS is present. A little bit of HbF is present, but HbA is not. It is in very, very less quantities. It is not present. That is what makes this sickle cell anemia to be homozygous and is a severe condition. Okay, you will have severe symptoms. Okay, so given that there is abnormal migration of hemoglobin, so you have a specific separate band in case of sickle cell only. Is it not? Be it sickle cell trait and sickle cell anemia, there is specific characteristic band in the sickle cell. So now what you can think of is here, there is an hemoglobinopathy, but you can attribute this to sickle cell. So for all those who have answered it right, yes, it is sickle cell. Yes, sickle cell. And what is, they are not asking you for the diagnosis here. What is the type of mutation here is only they are asking. What is the type of mutation here? Yes, what is the type of mutation? Yes, I'm waiting for the answer. Yes, it is going to be point mutation. Is it not? The type of mutation is going to be point mutation. And what point mutation? It is causing symptoms. If it is causing symptoms, so it is a missense mutation. So be it here, he is a 55-year-old person and he is not going for any, his SpO2 is normal and also given that you can think of the sickle cell as a trait and not anemia. Okay, sickle cell trait. Because it's a milder symptom you have. So this is a point mutation or missense point mutation. So what is a point mutation in sickle cell? Yes, what is a point mutation in sickle cell? In beta globin gene. In the 6th position. Yes, in the 6th position, what do you find? Glutamic acid gets converted to valine. Yes, glutamic acid gets converted to valine. This wild type is the normal one. Okay. Normal hemoglobin in mRNA, you have GAG and that is going to code for glutamate. But in the mutated one, there is a point mutation. Point mutation means what? A single nucleotide is what is changed. So here this A is getting converted to U here, because of which there is valine. Yes, so that is what is called as the sickle cell hemoglobin or the HB. Yes. Understood? So this is what is the type of mutation that you find in sickle cell, right? And what are the other questions they have asked is, classify mutation based on the effects of mutation on nucleotide sequence, on amino acid sequence, and based on function of the protein, and give suitable examples. So we will see that. Classification based on nucleotide sequence. Yes. So based on nucleotide sequence means what? First thing, what did we see now? Is the point mutation. Point mutation means point. Point means what? Only one nucleotide base is changed here, because of which abnormal thing has happened. Yes. So change in a single nucleotide base is what is the point mutation. Okay, base in a, change in a single nucleotide base. An example is what? Sickle cell anemia. Right? So that's what we saw. Then next is insertion. What is insertion? As the name says, insertion means there is one or more nucleotides that have been inserted in between the normal one. Okay, so there is addition of one or more nucleotide bases. That is what is insertion. Example is Tay-Sachs disease. Right? So Tay-Sachs, as you can see, look at this. Tay-Sachs means what? Hexosaminidase gene is what is affected here, right? So hexosaminidase normal gene, look at the sequence here, TCC-TAT. Okay. Now, Tay-Sachs gene is looking like this. There is insertion of this TATC in between. This is not at all present in the normal one, right? Rest of the thing is looking like TCC-CTA. TCC-CTA. Sorry, TCC-TA. TCC-TA is there. In between TATC is what is the additional nucleotides, is it not? So this has been inserted. So 4 nucleotides are being inserted in between. That is what in Tay-Sachs it happens. So it is an example for insertion. Next type is deletion. What is deletion? So in between one or more nucleotides are being deleted. That is what is deletion. Example is cystic fibrosis, right? Example is cystic fibrosis. So you look at this. Look at the sequence. You can see the 508 position is phenylalanine, which is CTT. Okay, normally. But in the CFTR gene. So in cystic fibrosis, what happens? In cystic fibrosis, the CFTR gene is what is mutated. How is it getting mutated? This itself, the CTT itself is not here. It should have been here, right? It's not here. So that is what this deletion. So 3 nucleotides are deleted. So one codon is deleted. So at the 508 position. So that is what happens in cystic fibrosis, and it's an example of deletion. Duplication means what? A segment of DNA is copied and inserted. That is duplicated. Right? So as look at this picture, you can see, you'll be able to find out this B gene is what is duplicated and inserted in between. So this is duplication. An example for this is going to be Charcot-Marie tooth diseases, duplication. Inversion means what? A segment is removed and it is attaching to the same chromosome, but now in a reverse fashion it is getting attached. Right? For example, I have this segment 1, 2, 3, 4. This is how my nucleotides are arranged. Now, what happens? This portion is getting removed, okay, and it is coming back and attaching to the same chromosome, but now in a reversed fashion, 4, 3, 2, 1. That is what is inversion. So look at this. B, C, D, E is there, but here E, D, C, B. That's how it's getting attached. That is what is inversion. Right? Some hemophilia A cases can show this inversion. Next is frame shift mutation, which is again important. So as we saw, there is no overlapping, there is no punctuation. These are the characteristics of genetic code, right? So what should happen? That is the characteristic of the genetic code. But what happens when some insertion or deletion has happened in that gene? Yes, look at this Tay-Sachs. Now, 4 base pairs are getting inserted here. So now what happens? Look at the amino acid sequence in a normal hexosaminidase gene. There is serine followed by tyrosine. But as in Tay-Sachs gene, since there is insertion of this 4 base pairs, what has now happened? TCT that's coding for serine, okay, fine. ATC that goes for isoleucine is coming in between. Then CTA that's coding for leucine. But look at this, look at this arrangement. Serine, tyrosine, glycine, proline, aspartate. That's how the sequence of amino acids should be. But since because 4 base pairs are now inserted here, the codon is read in a different manner and different amino acids are getting attached. So you find serine, isoleucine, leucine, cysteine and proline, which is not at all similar to that of the wild type gene, right? That is because just 4 base pairs, you can't just neglect this four base pairs and other amino acids can be normal. You can't think of that like that, right? So in the sequence, the codon has to be read 1, 2, 3, 4, 5. When something is getting inserted or something is getting deleted in between, the remaining of this line is going to be affected as a whole. As a whole polypeptide, it is going to get affected. So entirely different codon will entirely different polypeptide you will get. So that is what becomes a non-functional protein. Clear? So this is frame shift. So the reading itself is shifted. So this is what is called as the frame shift mutation. So insertion or deletion that has happened. So it shifts the reading frame. Okay, not in multiples of 3. Right? So when one codon alone is deleted, that codon alone will be abnormal, but rest of the codons will be coding for the same amino acid. So that can go like that. So look at the cystic fibrosis, only phenylalanine is not there, but rest of the amino acids are the same. Glycine, valine, and the other things will be the same, even after this deleted component. But in Tay-Sachs, this is not in multiples of 3. Here, not just one codon is removed. Here, 4 nucleotides are inserted in between. So the entire frame is getting shifted. Okay, the entire frame in which it is read itself is shifted, right? So that is what is called as the frame shift mutation. Again, the example for this is Tay-Sachs. Right? So these are the different classification based on the nucleotide sequence. Now, based on amino acid sequence. What is this based on amino acid sequence? See, we have silent mutation, missense mutation, and then nonsense mutation. Okay, what is silent mutation? So side by side, we are seeing the examples. They have asked, give various, explain with examples the separate question, but side by side, we are seeing with the examples. Okay, so that also just noted. So silent mutation means what? Look, silent mutation means what? Suppose we know, no, one amino acid will have 2 or 3 codons. So even if mutation has happened and the codon is changed, but still that codon is coding for the same amino acid, then you are escaped. Is it not? So CUA is there. If it gets mutated to CUC, both of which are coding for leucine, so the amino acid sequence is not going to be changed or the polypeptide is not going to be changed. So that is when you won't have any symptoms. So the mutation is silent and has no effect on the phenotype. So the mutation is silent here. That's why it is silent mutation. Missense mutation, again, you have 3 types: acceptable, partially acceptable, and unacceptable. Okay, acceptable, as you see, look, with the help of examples, we will see. Here, AUA is getting converted to AAU. Again, point mutation is what has happened here. But the amino acid is different here. Not in case of sickle cell. Here, the amino acid is going to be different. So, lysine is getting converted to asparagine. Now, what has happened? Adult hemoglobin, HbA, is getting converted to HB Hikari. But still, the patient is not having that much symptoms. Phenotypically or functionally, they don't have that much complications. So maybe, okay, fine, you can accept it. So it is an acceptable missense mutation. Right? Partially acceptable means what? GAA to GUA. Glutamate to valine. What is this? HbA to HB? Yes. Right? So if a patient has HBS, of course, he will have symptoms, right? But still, HBS, if it is a heterozygous condition, wherein you have both the adult hemoglobin and the sickle cell, it is called sickle cell trait. Right? In that case, only when the patient is exposed to deoxygenated state, he will have severe symptoms, but otherwise he can live normally. So that is why you call it as partially acceptable missense mutation. But unacceptable means what? It's incompatible with life. So there is a point mutation wherein histidine is getting converted to tyrosine. So HbA gets converted to a Boston hemoglobin or HB. So this is an unacceptable missense mutation. So here it becomes the protein itself becomes non-functional. So hemoglobin itself becomes non-functional. Partially acceptable, hemoglobin is functional only. But in unacceptable, the hemoglobin itself becomes non-functional and it is incompatible with normal life. Okay, so here in missense mutation, one amino acid is getting converted to another amino acid. That is what is missense mutation. Silent mutation means there is point mutation, there is one nucleotide change, but the amino acid is the same. That is what is silent mutation. Now coming to nonsense mutation. Nonsense mutation means one codon is getting converted to a stop codon. So now what happens when in a mRNA, when it is getting translated, if still it has to be translated, what happens when this codon gets converted to a stop codon in between? Yes, the protein translation gets stopped at that point. So you will have a truncated protein, right? Or another thing what can happen is, suppose this stop codon is getting converted to, stop codon is mutated, it becomes another amino acid and it is going on prolonging. So you will have another non-functional, different protein. Yes. So these two are the ways by which they can happen. Nonsense mutation can happen. So nonsense mutation, again, it's a point mutation where the codon gets converted to a stop codon. So there is premature termination of this translation process. So you will have a truncated non-functional protein. And the example for this is going to be beta thalassemia, severe form. Here there is this codon 39 wherein CAG is getting converted to a stop codon, right? So this mutate to a mutated codon. Yes, UAG. So, sometimes, U and T, they will write it like this. They will write it irreversibly, thinking of DNA. Okay, but still, we will understand it as stop codon. So, UAG, which is a stop codon. So, the effect, what has happened in between the protein synthesis will stop. Translation stops prematurely at this codon 39. So non-functional beta globin chain will be produced. So it's non-functional, right? So that is what is resulting in beta thalassemia. Or this terminator codon is altered to a coding codon. So that becomes a run-on polypeptide. So this is also possible, right? So, but basically nonsense mutation is one codon getting converted to a stop codon and you will get a truncated non-functional protein. That is nonsense mutation. So these are the types of mutations based on your amino acid sequence. All right. Next, classification based on your protein function. So function of your protein. Loss of function. Yes, the protein, if it is the function of the protein is lost. As in case of P53 mutation. Yes, if it is lost. Then gain of function mutation, when the protein function is aggravated or it is very much increased, it becomes more reactive. Yes. So again, cancers, you have EGFR mutations, wherein there is cell division is happening. It is not controlled. Is it not? Again, the control mechanism is lost in case of P53 mutation, that is loss of function. When the control mechanism, the one that when it is getting increased, the one that is going to stimulate the cell division is going to be increased, then that is what is gain of function mutation. Then dominant negative means the mutated protein that will interfere with the function of a normal protein. You have in osteogenesis imperfecta mutant collagen. Lethal mutation, the mutation that leads to death of an organism, that is what is lethal mutation. So embryonically, if there are lethal mutations, it will lead to death of the fetus. Then conditional mutation. Conditional mutation means what? The mutations will happen, or it expresses, or mutations are there, but it expresses only under specific conditions. Like for example, antibiotics. If you're going to grow bacteria in an antibiotic environment, or if you are giving antibiotics for a longer period of time, the bacteria will become resistant to the antibiotic, right? So over a period of time, it is as a it is becoming resistant to the antibiotic. So this drug resistant tuberculosis is an example for it, right? So in this case, what you will do, you will give one drug, you will give two drugs in combination, not just one drug. You will give combinations of drugs so that this resistance doesn't affect the treatment of the patient. Okay, so this is conditional mutation, means only under certain conditions this is expressed, mutations are expressed, right? So this is all about your classification based on protein function, right? So with this we will stop. So we have seen about the different types of mutation. Yes, hope this session was useful. So we will stop with this. We will see in the next session, right?